Namaskara Karnataka, welcome all of you to Vedantu Karnataka. So students, part 1 already sexual reproduction and flowering plants agide. So this is for all my CBSE students because I know, class 12 starts and you do not understand this chapter, then the whole chapter becomes a problem, then the continuation of next chapters also become a problem. So each channel, Ali Vedantu Karnataka channel, all the students around the country can watch this for 12th CBSE because this will have every single thing, and especially biology is going to be taken very thoroughly. Even if you're a NEET student, we will have some additional points for all of you. So I want all of you to subscribe to the channel if you're new and stay tuned and quickly smash that like button. Many of you are not liking the video, many of you are taking knowledge. So if you're taking knowledge, you should also give some love and respect to the teachers, right? So students, today we are going to do part 2 of sexual reproduction and flowering plants. So idalli mainly ನಾವು megasporogenesis ಬಗ್ಗೆ ಮಾತಾಡ್ತೀವಿ because microsporogenesis already last class ಅಲ್ಲಿ ನಾನು ಕಂಪ್ಲೀಟ್ ಮಾಡಿದ್ದೀನಿ. So ಇವತ್ತು ನಾವು pistil carpel ಬಗ್ಗೆ ಮಾತಾಡ್ತೀವಿ, ಓಕೆನಾ? So I hope all of you are ready. Bega video na like ಮಾಡಿ and also let me know if you're all enjoying the class as well. Okay, so students, start ಮಾಡೋಣ. First, NEET students ಗೆ ಈ chapter ಬಂದ್ಬಿಟ್ಟು almost 20 marks chapter because this chapter has around 5 questions. Okay, so 5 into 4 20 marks chapter idu. So it's one of the high weighted chapters. Now talking about pistil and carpel. So now first class ali entire flower we have learned and understood, and our flower ali I told you that there are 2 essential parts, right? Adu nam parents tara, they are like our essential parts in our life, that is without them we cannot go forward, right? But we also have some non-essential part like our friends, colleagues, workmates, etc. because without them also we cannot complete our life. So non-essential part directly does not involve into your life making decisions or your marriage or your relationship or your parents and your financial situation. They do not directly interfere, but namaga avaru jote maatadbodu like ee tara nanagondu problem ide, so if they want to help they will help, but directly nav yaar jote maatadodu, parents jote, and parents talk to us, so they are become essential part, and this corolla and calyx become the non-essential part. That means petal and sepal becomes the non-essential part. So second essential part that you're going to study in this chapter is going to be pistil and carpel. Okay, pistil and carpel. So pistil and carpel first we will draw, after that I will explain. Okay, one second students, pen inda idella agalla. Okay, yeah. So look here, this is swollen portion, right? And then there is a portion like this, and again a swollen portion. Okay, yes. So this is your stigma style ovary which forms the pistil or your carpel. So inside this swollen part there is going to be your ovules. Okay, there is going to be ovules, like different kind of placentation can be there. Now I'm just drawing this. So students, the label marana, first you have what? You have stigma. First you have what? Stigma. Stigma andre where the pollen grains are going to land. Okay, where the pollen grains land, and there on landing position, like now we have helipads, so like that, where the pollen grains are going to come and land is going to be called the stigma. Okay, it's going to be called the stigma. Now look here, further we go down, you can see one long slender like structure. This is called the style. This is called what? Style. Style is basically a connect. Okay, it's just a connect between stigma and ovary. Okay, stigma and ovary is just a connect. Clear to all of you? Now if you come here, the swollen part, this has everything that is going to go forward. Okay, so it one but most important or most essential part anta helbodu because adolagade ovules irutte. Ovules will have your egg, which is your female gamete, which has to fertilize with the male gamete for entire fertilization to happen. Okay, so this is going to be your ovary. Okay, and this is going to be your ovules. Okay, this is going to be your ovules. So this entire thing, students, we can call it a pistil or a carpel. What can we call it? A pistil or a carpel. Okay, many of these will be there. Now if any of you have a hibiscus or a China rose, okay, near your home, you can go and see the stigma very clearly. You will be able to see everything very clearly. Now students, see carpel matte pistil alva, does it always have to be single? It's not necessary, but if they are single, so your carpels, okay, if they are single, they are going to be called monocarpellary, okay. If they are many, they are going to be called multicarpellary. Multicarpellary, okay. If they are one, they are called as monocarpellary. If they are many, they are called as multicarpellary. Now, if multicarpellary alli ಎರಡು options ಇದೆ students, ಅವರಿಗೆ fuse ಆಗಿರಬೇಕಾ ಇಲ್ಲ free ಆಗಿರಬೇಕಾ ಅಂತ ಒಂದು option ಇದೆ. So, if in multicarpellary, if they are going to be free, okay, if they are going to be free, we call it apocarpus. Apocarpus, free ಆಗಿದ್ರೆ, okay. And if they are going to be fused, ಅದನ್ನ ನಾವು syncarpus ಅಂತ ಕರಿತೀವಿ. Like sometimes in your phone they ask, no, is your Google account synced with your phone number? Is your PAN card and Aadhaar card synced together? Sync ಅಂದ್ರೆ ಏನು? Fused ಅಲ್ವಾ? So ಇಲ್ಲಿ fused, okay, fused, clear? So in multicarpellary, two options are there, free or fused, okay, free or fused, that is apocarpus or syncarpus. Free andre apo, okay, apo is free and syn is fused, okay, syn fused, that is why they tell, have you synced your account, have you synced with this person, so that means something like fused. So remember this. Now there is a way to remember the examples, okay, in your NCERT 2 examples are given, there is a way to remember this. Students, you can remember this like this, M-A-M, ma'am. Can you remember this? Now look here, M for multicarpellary, I am just writing short forms here, multicarpellary. A for apocarpus. Now M is the most important, the example, Michelia. What is it called? Michelia. Okay, M is Michelia. Clear? Now syncarpus, syncarpus example to remember, you remember M-S-P, okay. Again M for multi, okay, S for syncarpus, and P for the important example, papaver. Okay, full is papaver somniferum, or you can also remember poppy plant. Okay, poppy plant. So can you remember this much? So multicarpellary. One second, students. Yes. Multicarpellary, okay, it's going to have two options. One is going to be apo, one is going to be syn. That means one is free, one is fused. Apo example is going to be ma'am. Remember me, ma'am, okay. And syncarpus is going to be MSP, which is going to be multi. Again, syncarpus example is going to be papaver somniferum, or you can remember poppy plant, okay, or you can remember poppy plant. Now let's see NCERT, what NCERT has to say. So you'll notice, students, different diagrams I've given. All of you can see thalamus, above that you can see ovary, and then you can see style, and then you can see stigma. All of you would have seen the stigma in your you know, hibiscus plant, okay, in your hibiscus plant. Ivaga ee B option nodi, ideno syncarpus. Can you see ella stigma fuse agide, okay, entire stigma is fused, so that's why we call it syncarpus ovary, okay. Ill nodi ella free agide, itara free agide. Okay, so this is called as, the carpels are free, so we call this apocarpus. In example kottidare students nodi, papaver, okay, MSP, papaver, okay, papaver, that's the example for syncarpus. Il nodi a multicarpellary apocarpus, Michelia, so ma'am, okay, a diagrammatic representation of anatropous ovule, we will come into this. First, if you understood all of this, please quickly mark your attendance, okay, quickly mark your attendance. 1 second students, jacket haakbidtini. AC is not ACing, it's working differently today. So we should do our part, alva. ಏನು ಮಾಡೋಕ್ಕಾಗುತ್ತೆ, I can't scold the AC ಅಲ್ವಾ, sometimes ಈ ಥರ ಡ್ರಿಪ್ ಆಗುತ್ತೆ, ಏನು ಮಾಡೋಕ್ಕಾಗಲ್ಲ. Okay, yes. So we are settled. Now we can continue. Okay, next is going to be anatropous ovule. Okay, next is going to be anatropous ovule. Now students, anatropous ovule, ಏನದು ಹೆಸರು ಎಲ್ಲಿಂದ ಬಂದಿದೆ ಅಂತ we will first understand. Okay, students, ovary ಬಂದ್ಬಿಟ್ಟು different types ಇದೆ, but in case ಮೂರು parts ಇದೆ, funicle, chalazal, micropylarine. ಇದೆಲ್ಲ ಒಂದು ಲೈನಲ್ಲಿ ಬಂದರೆ different type of ovule ಅಂತ ಕರಿತೀವಿ. In case only chalazal and micropylar is in the same line and the funicle is a little this side, we call it anatropous. So depending on the shape, there are different types of ovaries, okay, like circle agidre, circulate ovary, other different types of ovaries are present. So now remember that you're going to study anatropous ovule, which is present in your most of the angiosperms. Clear? So let's first draw the anatropous ovule and then I will explain you each part of it. Okay? So students, ingu draw madbodu, ingu draw madbodu. Nan ee tara draw martini because nanage adu ಸ್ವಲ್ಪ easy explain ಮಾಡಕ್ಕೆ and draw ಮಾಡಕ್ಕೂ ಕೂಡ. So ee tara draw ಮಾಡಿ. Diagram keltare, okay? For both they might ask you the diagram. So learn and study now only. Okay, these are the, yes. If you look at this, this is the ovary, okay? This is the ovary. Now inside this, I am going to draw one oval structure. You have to tell me what is the oval structure. And around the oval structure, I am going to be drawing some cells. These are all very important, okay? You will understand as I am explaining. Don't worry. Okay, just now ishtu draw ಮಾಡಿದ್ವಿ, rest ಆಮೇಲೆ draw ಮಾಡೋಣ. So ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇದು ovule, okay? ಇದೇನು ovule? Anatropous ovule. So our diagram ಕೇಳ್ತಾರೆ boards ಅಲ್ಲಿ, directly they'll ask you draw the anatropous ovule. Now just look at this. You will be noticing that I have drawn two, like, you know, kind of like a foldings I have drawn. Those are nothing but integuments, okay? So this one, which is outside, okay, is called as outer integument. What is it called? Outer integument. Clear? Outer integument. What will be the inner one? Ma'am, obviously inner integument. Inner integument. Okay? Inner integument. Clear to all of you? Idu outer, idu inner. Color ಮಾಡಬೇಕಂದ್ರೆ different colors ಅಲ್ಲಿ color ಮಾಡೋಣ. ನಮಗೆ ಆಪ್ಷನ್ ಇದೆ ಸ್ಟೂಡೆಂಟ್ಸ್. ಕಲರ್ ಮಾಡೋಕೆ ಆಪ್ಷನ್ ಇದ್ರೆ ನಾವು ಮಾಡಬೇಕು. No. Okay, ಇದು ಔಟರ್ ಇಂಟೆಗ್ಯುಮೆಂಟ್. ಓಕೆ, ನನ್ನ ರಿಯಾಕ್ಷನ್ ರೆಕಾರ್ಡ್ ಮಾಡಿ ನೀವು ಪೋಸ್ಟ್ ಮಾಡಬೇಡಿ. ಓಕೆ, ಸೊ ಇದು ಇನ್ನರ್ ಇಂಟೆಗ್ಯುಮೆಂಟ್. ಒಂದು ಬೇರೆ ಕಲರಲ್ಲಿ ತೋರಿಸಿದ್ದೀನಿ, ಇನ್ನರ್ ಇಂಟೆಗ್ಯುಮೆಂಟ್. ಓಕೆ, okay, now look here, outer done, inner done. Now you will be thinking, ma'am, what have you drawn all this? Before that, wait students, ಈ ಸ್ಟಾಕ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ ನೋಡ್ತಾ ಇಲ್ವಾ, ಇದು ಬಂದ್ಬಿಟ್ಟು ಫ್ಯೂನಿಕಲ್. ಏನಿದು? ಫ್ಯೂನಿಕಲ್. I told you, that funicle, chalazal, micropylar comes in a straight line. We call it different types of ovule. So this is the funicle. Ee opening kansta ilva, here where the integuments are absent, this is called the micropyle. What is it called? The micropyle. Clear? Micropyle. Done? Clear to all of you? Is it clearly visible? Now, the portion opposite to the micropyle, okay, the portion opposite to the micropyle is called the chalazal end. Okay, chalazal end. Done? Clear. Now look here students, there's one very important spot here. If you notice properly, you will notice that there is a region that attaches funicle and the ovary together, the ovule together. This notch is called as hilum. This notch is called as hilum. Okay, understood? Now the pink color structure that all of you will be waiting, when will this ma'am actually label this? This is going to be called the embryo sac. What is this embryo sac? Inside that what will be there? The female gamete will be there. Okay. Now students, the green color cell banbit tumba important cells. Why? Because these are called as nucleus cells. Nucleus cells. Now if all of you can, go back and understand, or go back to the first part of this chapter, you will remember that I drew tapetal cells, right? For the microsporangia, I drew tapetal cells. Now what was tapetal cells doing? They were doing the nourishment that was required, right? Same way, in every step of the plant, there is going to be providing, someone will be providing nourishment for the growing pollen grains, for the growing embryo sac, for the growing embryo. So every step there is someone who is going to provide the nourishment. Here nucleus is the one who is going to provide the nourishment. Okay, listen to me carefully. So nucleus provides nourishment. Okay, everything's we will write the job. Okay, so nucleus provides nourishment, okay, for the growing embryo sac. Okay, now this is the embryo sac where this embryo sac is going to act as the female gametophyte. Okay, now how I told you pollen grain is the male gametophyte, this is going to be female gametophyte. Okay, female gametophyte. Now, inner integument, students, these two are going to help for only one job, that is going to be protection. Okay, protection ige help ಮಾಡ್ತಾರೆ. Now, micropyle end is the portion where the integuments are absent. Okay, portion, or we can say region. I think region would be a good word. Oh, what is that? Oh, 1 second, students. Region where integuments are absent. Okay, where integuments are absent. Clear? Now, funicle. What do you think funicle's job will be, ma'am? Very simple, ma'am. Funicle would be acting like a stalk. Yes, very correct. It is going to act like a stalk. Okay, stalk. Now, hilum, students, nan helidini, it's like a notch that connects, or that connects funicle and ovule. Okay, that's the region that connects the funicle and ovule. So, is the ovule clear to all of you? Just look at the ovule, and if any of you have a doubt, please let me know. So, you'll notice, students, illon stalk ತರ structure ಇದೆ, and if you come like this, you will see two integuments, which main function is going to be protection. Okay, main function is going to be protection. And now, if you look at this, you will notice that there is a region where the integuments are absent, that is called the micropylar region. Opposite to the micropylar region, there is a chalazal end. Okay, chalazal end. Now, once you see the embryo sac, around the embryo sac goes a set of nucleus cells, which are actually the cells that provide nourishment for the growing embryo. Okay, growing embryo, that is inside the embryo, what is the egg is there. Okay, so it will provide the nourishment. And then if you look here, there is a small notch like structure that attaches the funicle and the ovule together, which is called the hilum. Okay, which is called the hilum. And then everything is understood. Okay, this is clear, I hope. Okay, just let me know if any doubts are there. Now, students, what is exactly going to happen? Okay, now, ivaga, one thing that is going to happen is, let me just draw an ovule here again. Color change madana. Bright color beku. Okay, this is bright. Okay, now look here, all of you. Now, I am going to draw ovule. Yes, this is ovule, correct? Okay, I will try. One second, students. Okay, because this diagram multiple times repeat madbeko, so first we will draw it properly. Pen sari aagta illa. It will be like this ma'am is always complaining about board and pen. True, students. A teacher, if she doesn't get a good studio, no, she'll be very irritated the whole class. Okay, this is the ovule, correct? Now, in between, what are we going to draw? Embryo sac we'll draw. Okay, actually, this chapter is very interesting. Okay, I want all of you to listen, because my NEET students love this chapter. So that's why I'm telling you, you should listen to this chapter. Okay, this is an embryo sac, correct? Now, I told you there's one very important cells here. Okay, there's one very important cells here. Like this, I'm just drawing box, box, box. Okay. Like this box I have drawn. Okay, can you see this? Now, let's just okay. ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್, ನಾರ್ಮಲಿ ಇದೆ ನಮ್ಮ ಓವ್ಯೂಲ್ ಡಯಾಗ್ರಾಮ್ ಅಲ್ವಾ, ಇದೆ ನಮ್ಮ ಓವ್ಯೂಲ್ ಡಯಾಗ್ರಾಮ್. ಇಲ್ಲಿ ದಿಸ್ ಇಸ್ ಯೋರ್ ನ್ಯೂಸಿಲಸ್ ಸೆಲ್ಸ್, ರೈಟ್? ದಿಸ್ ಇಸ್ ಯೋರ್ ನ್ಯೂಸಿಲಸ್ ಸೆಲ್ಸ್. This will be the nucellus. Now, what happens? One of the nucellus cells, okay? One of the nucellus cells, that is this cell, let's assume, one of the cells become very prominent, and he will be like, wait, I want to become the megaspore mother cell. So, this fellow will end up forming megaspore mother cell. What are we doing? Megasporogenesis, correct? So we are going to get our MMC, which is diploid. Okay, one of the nucleus cells will be like, I want to become the megaspore mother cell, and they will come forward. And now what is going to happen, students? You'll nodi. Ida mele definitely meiosis agutalva. Yes, ma'am, meiosis agutte. So meiosis admele nam microsporogenesis alli enaito. Meiosis admele microspore mother cell inandre pollen mother cell underwent meiosis. Adamele we got microspore tetrad. Illu somewhat similar ಆಗ್ತಾ ಇದೆ, but ಸ್ವಲ್ಪ difference ಇದೆ. ಇಲ್ಲೇನಾಗುತ್ತೆ students ನೋಡಿ, here we get, okay, here also you can see that we have got a megaspore tetrad. Correct? You can see that we have got megaspore tetrads. But a small problem ಇದೆ. ಈ tetrad condition ಅಲ್ಲಿ, okay, we have got, ಇಲ್ಲಿ ಬರೀತೀನಿ, ಈ megaspore tetrad ಅಲ್ಲಿ students ಎಲ್ಲ survive ಆಗಲ್ಲ. Everything will not survive this. What is going to happen here? Remember carefully that out of the 4 of them, 3 of them are going to degenerate. 3 of them are going to become what? They are going to degenerate. Okay, these 3 are going to disintegrate or degenerate. Okay, degenerate. The only person who is going to survive this, okay, the only person who is going to survive this is going to be the one that is near the chalazal end. This is important for NEET point of view. The chalazal end, okay, megaspore will be surviving, and because only one megaspore is surviving, ನಾಲ್ಕಿತ್ತಲ್ಲ ಅದರಿಂದ only one is surviving. So ಅದಕ್ಕೆ ನಾವು ಈ development ಗೆ ಏನು ಹೇಳ್ತೀವಿ? We call this development as monosporic development. Mono means one, spore means one spore, okay, monosporic development. Monosporic development. That means out of the 4, 3 degenerated or disintegrated and only one became functional. Only the one that was near the chalazal end became functional spore. Functional megaspore. Functional megaspore. This kind of development is called as monosporic development. This kind of development is called as what? Monosporic development. Clear to all of you? Now students, that one fellow is going to undergo multiple divisions, okay? That fellow is going to do many mitosis, okay? Dia to 4, 4 to 8. This kind of clarity you need while studying biology, okay? Not just learn 1 or 2 things and be like, oh, I know biology. That's not how you know biology, okay? That one fellow student is going to undergo multiple mitotic divisions, okay? To be specific, 3 mitotic divisions. Okay, 3 mitotic divisions. We can say specifically karyokinesis happened, okay? 3 mitotic divisions, and then what are we going to get from 1? We are going to get 2. Okay, so I'll start from 2 stage. Okay, I just wanted to show you this 1. So from 1 we will start 2, right? So we will just go back. Okay, we'll start from 2. 2 to what will happen? 4 stage will happen. Okay, 2 to 4. 4 to what? Obviously, ma'am, 8 will happen, right? 4 to 8 will happen. So here 8 you will start seeing. Okay. Like this. This is how you are going to see the embryo sac. So here you will notice that every step these people are going to undergo a mitotic division, and ultimately we are going to get a 8 nucleated stage. Yes or no? All of you agree to this? Anyone who does not agree to this, I would want to know why you are not agreeing to it. Okay, now look here. Okay, 3 mitotic. All this notes, students, ನಿಮಗೆ ಈ notes ಎಲ್ಲ ವೇದಾಂತು ಕರ್ನಾಟಕ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲಲ್ಲಿ ಇರುತ್ತೆ. ಮತ್ತೆ ಗೋಪಿಕಾ ಬಯಾಲಜಿ official ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಫಾಲೋ ಮಾಡಿದ್ರೆ ನೀಟ್ ಕ್ವೆಶನ್ಸ್, ನೀಟ್ ನೋಟ್ಸ್ ಎಲ್ಲ ಅವೈಲೇಬಲ್ ಆಗಿರುತ್ತೆ. ಸೊ ಖಂಡಿತ ಫಾಲೋ ಮಾಡಿ. ಓಕೆನಾ? ದಟ್ ಇಸ್ ದ ಓನ್ಲಿ ಪ್ಲೇಸ್ ವೇರ್ ಐ ಕ್ಯಾನ್ ಸ್ಟೇ ಕನೆಕ್ಟೆಡ್ ಟು ಯು. ಓಕೆ, so this is done. 3 mitotic divisions are done. Now students, we are going to draw the embryo sac. Illu ನಾನು ಇದು remove ಮಾಡ್ತೀನಿ so that I can copy paste easily. Okay. Okay, we are going to learn embryo sac. Okay, done. Embryo sac. Now listen to me carefully. So students, namaga 8 cell stage siktalva. So illi nodi. Oh, 1 second. Yes, ma'am excited about things that ma'am has not seen. Okay, nice, no? What do you guys think? Okay, enough. Okay, close. Okay, so nice, no? Okay, so here, okay, this was the stage we got, right? Illi enittu students, 8 we got. Okay, 8 nucleate. Okay, 1 2 3 and 4. Ivaga enagutte, from this stage we are going to get an embryo sac stage. Okay, from this stage we are going to get an embryo sac stage, and this is one of the most important things that you have to learn as well. Listen to me carefully how this entire thing is going to happen. So students, ee stage inda ee stage ge on complete movement agutte. That means these people will start moving towards the pole. Who will start moving towards the pole? I'll show you. Wait. Now look here. To this side, you will start seeing 3 antipodal cells. To one side of the, the embryo sac, you will see this antipodal cells moving towards one pole, and here you will get 3 antipodal cells. What will you get? 3 antipodal cells. Okay, here on the top. Now, to the bottom also some people will be running. Okay, that is going to be 1 synergid, 1 more synergid. Okay, and right in the center we are going to have our egg. Okay, egg that is going to fuse with the male gamete. Okay, egg is going to be there. Now to the center, students, 2 people will be like, no, I want to be the center person, so they will become the polar nuclei. Polar nuclei. Now we also have one more cell which is called the central cell. Okay, which is called the central cell, which will be going around like this. We usually draw it like this, or we can shade it with different color also, like this. Okay, this is the central cell. So this is your embryo sac. Okay, now you can open NCERT and see if this is your embryo sac. So ee 3 cells again karitivi navu adanna antipodal cells anta karitivi. Antipodal cells. Okay, now you will be thinking, ma'am, ee antipodal cells, which side did they run to? Students, they ran to the chalazal end. They ran to the chalazal end. So how will you remember this? AC anta remember madi. Enanta remember madi? Antipodal cells, chalazal end. Okay, antipodal cells, chalazal end. So AC anta remember madi. Okay, now these 2 cells are going to be called the polar nuclei. What are they going to be called? Polar nuclei. They are considered as a single cell. Okay, they are considered as a single cell. You'll notice, students, these 2 blue color structures. Okay, these 2 blue color structures are going to be called as synergids. Okay, and right at the center we have our egg. Okay, and this green color thing is called the central cell. Now on the synergids there are some specialized structures. On the synergid there are some specialized structures which are called as filiform apparatus. What are they called? Filiform apparatus. Ee filiform apparatus ige on dodda job ide students, ivagalla fertilization time alli. So these are called your filiform apparatus. Filiform apparatus. Done? Clear? This is your embryo sac. Okay, so here look here, 1, 2, 3 cells. Okay, 4, 5, 6, 7. So this embryo sac will be called 7-celled. But how many nucleus? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleated. 7-celled, 8 nucleated embryo sac. This question tumba time repeat agutte because it's ಸ್ವಲ್ಪ confusing alva. So always remember, cell on kammi irutte, nucleus on jasti irutte, because ee cell na we consider as one single cell. Polar nucleus is considered as one single cell. Okay, even though they have separate nucleus, because here we got 8 nucleated stage. Okay, now clear to all of you? Students, clear to all of you? Yes or no? This is what has happened. Okay, 7-celled, 8 nucleated embryo sac we have formed. Now inside this embryo sac, what do we see? We see the egg. Okay, one second. Yes. Yes, done. Okay, this is very, very important. Very, very important. So I hope this is clear to all of you. Yes or no? Okay, done. Now embryo sac is done. Now we will go through NCERT. So here megasporogenesis, illon heading bariyana students. Okay, illa andre you many of them will be thinking, yo, what did this ma'am teach and go? Megasporogenesis. Okay, one second. Megasporogenesis, no? Okay, one second. People keep writing by then ma'am will fix the heading. Let's see if we'll write correct. Yes, megasporogenesis. Okay. Here we got change ಆಗ್ತಾಯಿಲ್ಲ. Yo, what happened? Got stuck, I guess. One second. Let's try now. Some problem happened, no? You people put nazar, that you ma'am was so excited, so it went wrong. Okay, megasporogenesis. Clear? Done? Okay. So sorry I took some time off yours. This is one topic that we have completed, and now we have learned the embryo sac as well. Now let's go through NCERT. Okay, so none class will NCERT separate, or the you don't have to take time, because I'll be making you read NCERT as well. Okay, now the megasporangium, that is the ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. So we are talking about typical angiosperm ovule, which is very important for you to know. Okay, now the ovule is small structure attached to the placenta. Now placenta, I know some of you might not know. Ill nodi, this is your ovule. Okay, this is your ovule. Sorry, this is your ovary. Ee ovary olagade ovule ee tara attach aguttalva. So like this. This becomes the placenta. Okay, this region is the placenta, where the ovule is going to attach to the ovary. Okay, placenta by means of stalk. Can you see the stalk? The funicle. Okay, so the stalk is called the funicle. Now the body of the ovule fuses with the funicle. Very, very important. The body of the ovule fuses with the funicle in a region called as hilum. Okay, now Thus, hilum represents the junction between ovule and funicle. So, ataranu baribodu that hilum is a junction between ovule and funicle, or you can write hilum attaches funicle to the ovule. Okay. Now, each ovule has 1 or 2 protective envelopes. Okay. Each ovule has 1 or 2 protective envelopes called as integuments. Integuments encircle the nucellus except at the tip where there is a small opening called as micropyle. Okay, there's a small opening where the integuments are absent, that is called the micropyle. Okay, and now it's organized. Opposite the micropylar end is the chalazal end. Opposite to the micropylar is the chalazal end. I told you this, opposite to micropylar is the chalazal end, representing the basal part of the ovule. So, who represents the basal part of the ovule? The chalazal end. Okay, so micropylar end top and bandre chalazal basal part agutte. Okay, enclosed within the integuments is a mass of cells. This is important. Okay, enclosed within the integument is a mass of cell called as nucellus. Now, cells of nucellus have abundant food reserve. See, cells of nucellus will have abundant food reserve. Why, students? Because they are the one who is providing the nourishment. If I am giving nourishment to you, that means I am giving food to you. I should have more food, no? I cannot have only this much food and be like, okay, you take from my plate. So, more food reserve should be there, okay? Located in the nucellus is the embryo sac or the female gametophyte. And ovule generally has a single embryo sac formed from a megaspore. Now, look here, megasporogenesis. The process of formation of megaspores from megaspore mother cell. From what? From megaspore mother cell. Megaspore mother cell is called as megasporogenesis. So, microsporogenesis enito the process of formation of microspores from microspore mother cell or pollen mother cell with the help of meiosis is called as microsporogenesis. Atara illi megasporogenesis ಬಗ್ಗೆ ಮಾತಾಡ್ತಾ ಇದ್ದೀವಿ. Okay. Now, ovules generally differentiate a single megaspore mother cell in the micropylar region. So, ಇಲ್ಲಿ ನೋಡಿ students, entire process explain ಮಾಡಿದ್ದಾರೆ. First, we will finish reading NCERT. Okay, that's all. NCERT main portions I only have taken. Will nodi megaspore mother cell, nucellus, micropylar end. Okay, you can see from the nucellus that is present in the micropylar end will start becoming the megaspore mother cell. Allinda illi enagutte nodi division agutte. Megaspore dyad formation. Dyad andre 2. Okay, allinda tetrad formation agutte 4. But unfortunately, 4L only one will be able to survive. That one fellow will go through a dyad phase. Okay, then he will go to a tetrad phase. From tetrad phase, he will go to an octate phase, that is 8 nucleated stage, and then you will be able to see antipodal cells, 2 polar nuclei, central cell, egg, synergids, all. So, synergids will be towards the micropylar end. Okay, ನಾನು ಉಲ್ಟಾ ಡ್ರಾ ಮಾಡಿದ್ದೀನಿ, ಇವರು ಈ ಥರ ಡ್ರಾ ಮಾಡಿದ್ದಾರೆ. I have basically drawn this. Okay, ಇಲ್ಲಿ ನೋಡಿ students, filiform apparatus, synergids, egg, central cell, polar nuclei, antipodals to the chalazal end. Okay, remember like this, AC. Okay, AC. A for antipodal, C for chalazal end. Okay, AC is how you will remember this. Okay, I hope it's clear. Illon yolk tara they have drawn is the polar nuclei. Okay, what they have drawn is like yolk. Okay, so embryo sac is understood. Now what is our job? Now we have formed male gamete and female gamete. Now our task is to make sure that these two meet each other, right? We have to make sure that these two people meet each other. Now for making them meet, we have to go through a process which is called as pollination. Pollination andre in students simple. Ma'am, transfer of pollen grains from the anther to the stigma. Antheral pollen grains ide, stigma the landing place ide alva. So those students who forgot about it, illond recap madana bega. So idu nam filament. And this is our anther. What does the anther have? It has a lot of pollen grains. Okay, inside lot of pollen grains will be there inside the pollen sac. After dehiscence, okay, this line dehiscent admele, the pollen grains will start releasing. Okay, pollen grains will get released. Now what do we have here? We have the one and only stigma. Okay, we have one and only stigma. Now, this stigma is what, students? It is the landing place, right? The stigma is going to land here. Sorry, the pollen grains are going to land on the stigma. Now, for this process only, we call pollination. What is happening here? Look at the process. Here, what is happening? Transfer is happening. Transfer of whom is happening? Transfer of pollen grains transfer of pollen grains from anther to stigma. Anther to stigma. So transfer of pollen grains from anther to the stigma of same plant irbodu illandre different plant irbodu. Okay, same plant irbodu illandre same plant alli eradu flowers agirbodu. So athara more categories of pollination ನೀವು ಓದಬೇಕಿಲ್ಲಿ. Okay, now ನೋಡಿ, so this is your anther. So idanna full we call it stamen. Okay, we cannot just write anther students. Label ಮಾಡೋ ಟೈಮಲ್ಲಿ ಕರೆಕ್ಟ್ ಆಗಿ ಲೇಬಲ್ ಮಾಡಬೇಕು. This is filament ಅಂತ ಬರಿಬೇಕು. Okay, this should be written as this is filament and this should be written as this is anther. Okay, and together you always call it stamen. You cannot write just anther. Here you cannot call the only stigma, you cannot be highlighting, you have to say entire thing is carpel. Okay, entire thing is carpel. Now look here, depending on the types, you have two types of pollination. Okay, two types of pollination. Okay, look here, first type, second and third type. I'll be explaining this with the help of a diagram as well. First we will try to learn what is the terms. Autogamy. Autogamy. Students, auto mode andre eno? Auto mode andre eno? It is a situation, or we set it in the phone in such a way that contrast, brightness, exposure ella automatically set agutte. So autogamy andre self pollination. Okay, autogamy andre eno? Self pollination. In simple words, self pollination you would have studied in smaller classes in EVS. Okay, self pollination, or in science. Okay, second one students, geitonogamy. You wouldn't have studied this. Geitonogamy, the term might look like, oh my god, what is this geitonogamy? But let me tell you, it is not such a big word. I will teach you the word. Okay, I'll teach you what is the meaning of this. And then we have xenogamy. Xenogamy students, nimigella gottu, generally we call xenogamy as cross pollination. We call it cross pollination. Okay, now let's try to learn all of these terms with the help of, you know, diagrams. Okay, with the help of a diagram we will try to explain or understand these terms. Okay, so yeah, kinds of pollination. Nanu illu idu remove martini, so I can copy paste the flower. I hope all of you have liked the video and you're going to share with your friends as well. Okay, don't be conjuice. Okay, ready? Kinds of pollination. Okay, now look here. First I told you autogamy. Okay, we will learn it. We will learn it with the help of diagram. Okay, autogamy andre eno? Exactly ಏನು ನಡೀತಾ ಇದೆ autogamy ಅಲ್ಲಿ? As I told you, students, first understand it's transfer of pollen grains from anther to the stigma of a same flower. Okay, same flower. So andre ಇಲ್ಲಿ ನೋಡಿ, this is a stem. Okay, we have two flowers here. So look here, here we'll have, okay, what is this? Stigma, correct? From here what do you see arising? Anther. Here also what do you see? Anther. Okay, now here you will have a small swollen portion. like this swollen portion, and from here you will see petals coming. Okay, like this one flower you can see, correct? Yes or no? Now look here. Okay, this is the flower. Now what is going to happen, students? The pollen grains from the anther is going to fall onto the stigma. This is called as self-pollination. Yes or no? This is called as self-pollination. Yes, ma'am, this is called as self-pollination. That means here what is happening? Here basically transfer is happening on the same flower. Okay, same flower. That is why we call it self-pollination. We call it what? Self-pollination. That means transfer is happening. So, pollination definition one bit similar agirutte. That is transfer of pollen grains from anther to stigma. Idu same definition but stigma idu anther to stigma of same flower irutte, different flower irutte, different plant irutte, only that kind of difference will be there. Okay, only that kind of difference will be there. Clear to all of you? So let's just bring it here. Now here also you can see a flower. Now what is going to happen, students? I don't know if I can stick it here. Now here also let's draw a flower. Okay, this is a flower. So here also anther will be there. Yes, ma'am, anther will be there. Okay. Now here, what do you see? One second. Okay, so here what's happening, students? One plant, yes ma'am, one plant. Here the anther is going to go from here, sorry, the pollen grains are going to go from here to here. That means on the same plant, okay, on the same plant but different flower. Same plant, but different flower. Same plant but different flower. Clear to all of you? Same plant but different flower. So, transfer of pollen grains from anther to stigma of the same plant but different flower is going to be your definition. Okay. Now, last one is going to be cross pollination. So, students, idike ನಾವು ಏನು ಕರಿತೀವಿ gaietnogamy. Okay, gaietnogamy. Now, here we call it cross pollination or xenogamy. Okay, xenogamy. Xenogamy. Now look here. Here we have one plant, one another plant. Okay, so from here pollens are falling here. Okay, so this is completely different. Okay, completely different. This is one different. This is different. Okay, different plant completely. So cross pollination means different plant. So, transfer of pollen grains from anther to stigma of different plants. Clear? So, that is called as cross pollination. So, students, illi ಮೇನ್ ಆಗಿ ನಿಮಗೆ ವಾಟ್ ಯು ಶುಡ್ ರಿಮೆಂಬರ್ ಇಸ್ ದ್ಯಾಟ್ ಈ ಎರಡು ಸಿಚುವೇಶನಲ್ಲೂ ಅಂದರೆ ಸೇಮ್ ಆಟೋಗ್ಯಾಮಿಯಲ್ಲೂ ಗೇಟ್ನೋಗ್ಯಾಮಿಯಲ್ಲೂ ವೇರಿಯೇಷನ್ ಇರಲ್ಲ, ನೋ ವೇರಿಯೇಷನ್, ಬಿಕಾಸ್ ಇಟ್ ವಿಲ್ ಬಿ ಲೈಕ್ ಕ್ಲೋನ್ಸ್ ಅಲ್ವಾ, ಕ್ಲೋನ್ಸ್ ಥರ ಇರುತ್ತೆ. ಸೊ ನೋ ವೇರಿಯೇಷನ್. ಎರಡು ಸಿಚುವೇಶನಲ್ಲೂ ವೇರಿಯೇಷನ್ ಇರಲ್ಲ. ವೇರಿಯೇಷನ್ ಇಲ್ಲ ಅಂದರೆ ಪ್ಲಾಂಟ್ಸ್ ಕೆನಾಟ್ ಸರ್ವೈವ್ ಫಾರ್ ಅ ಲಾಂಗರ್ ಟೈಮ್. ಓಕೆ, ಬಟ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನಲ್ಲಿ ದೇರ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ವೇರಿಯೇಷನ್. Cross pollination, there is going to be variation. Clear to all of you? Yes or no? I hope this is clear. Yes, perfect. Ouch, let me just sit for a minute. Transfer of pollen grains from anther to stigma, autogamy, geitonogamy and xenogamy. Okay, autogamy means what, students? Same flower. Okay, anther to stigma of same flower. Geitonogamy andre eno? ಸೇಮ್ ಪ್ಲಾಂಟ್ ಬಟ್ ಡಿಫ್ರೆಂಟ್ ಫ್ಲವರ್. ಒಂದೇ ಪ್ಲಾಂಟಲ್ಲಿ 3-4 ಫ್ಲವರ್ ಇದ್ದರೆ ಡಿಫ್ರೆಂಟ್ ಫ್ಲವರ್ಸಲ್ಲಿ ಪಾಲಿನೇಷನ್ ನಡೆಯುತ್ತೆ. ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಅಂದರೆ ಏನು? ಕಂಪ್ಲೀಟ್ಲಿ ಡಿಫ್ರೆಂಟ್ ಪ್ಲಾಂಟ್ಸ್, ಮೇ ಬಿ ಸಿಮಿಲರ್ ಸ್ಪೀಶೀಸ್ ಇರುತ್ತೆ ಬಟ್ ಡಿಫ್ರೆಂಟ್ ಪ್ಲಾಂಟ್ಸಲ್ಲಿ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಆಗುತ್ತೆ. ನಾವು ಆಟೋಗ್ಯಾಮಿ ಮಧ್ಯೆ ಗೇಟ್ನೋಗ್ಯಾಮಿಯಲ್ಲಿ ವೇರಿಯೇಷನ್ ಇರೋದೇ ಇಲ್ಲ, ಬಿಕಾಸ್ ದೇ ಆರ್ ಕೈಂಡ್ ಆಫ್ ಫಾರ್ಮಿಂಗ್ ದ ಕ್ಲೋನ್ಸ್. ಇವಾಗ ನೀವು ಮದುವೆ ಮಾಡಿದ್ರೆ, ಲೈಕ್ ಇಫ್ ಯು ಆರ್ ಮ್ಯಾರಿಂಗ್ ಯುವರ್ ಫ್ಯಾಮಿಲಿ ಓನ್ಲಿ, ಅಂದರೆ ನೀವು ನಿಮ್ಮ ಫ್ಯಾಮಿಲಿ ಒಳಗಡೆನೇ ಮದುವೆ ಮಾಡಿದ್ರೆ ದೆನ್ ವಾಟ್ ಎವರ್ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸ್ ಇಸ್ ದೇರ್ ಅದೆಲ್ಲ ಕಂಟಿನ್ಯೂ ಆಗುತ್ತೆ ಅಲ್ವಾ. ಏನಾದರೂ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ ಇದೆ ಅದೆಲ್ಲ ಕಂಟಿನ್ಯೂ ಆಗುತ್ತೆ, ಬಿಕಾಸ್ ಯು ಆರ್ ಮ್ಯಾರಿಡ್ ಇನ್ಸೈಡ್ ದ ಫ್ಯಾಮಿಲಿ. ಬಟ್ ಇಮ್ಯಾಜಿನ್ ಯು ಆರ್ ಮ್ಯಾರಿಡ್ ಫ್ರಮ್ 2 ಡಿಫ್ರೆಂಟ್ ಫ್ಯಾಮಿಲೀಸ್. ಓಕೆ, ಅಂದರೆ ಸಮ್ ಕಂಪ್ಲೀಟ್ಲಿ ಸಪ್ರೇಟ್ ಫ್ಯಾಮಿಲಿ ಅಂದರೆ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸ್ ವಿಲ್ ನಾಟ್ ಬಿ ವೆರಿ ಪ್ರಾಮಿನೆಂಟ್. ಓಕೆ, ಸೊ ಫಾರ್ ದ ಜನರೇಷನ್ಸ್ ಟು ಕಮ್. But if you're marrying inside the family, everyone in the family have diabetics or they have gone through stages of cancer, then there's a high chance that your kid also will have it, because both of you are going to have the same characteristics, husband and wife. So adike it might be continued in your next generations as well. But when you're doing cross pollination, variation is more clear. So just like that only, like marrying the what do you tell for that, here in Karnataka that culture is not there, I guess. It's mainly seen more in Tamil Nadu and all. This one, Mama Ponnu, Mama Paiye. Okay, here I think it's very less comparatively. So that is the thing what I'm trying to say. Okay, clear to all of you? I hope it's clear. Now students, one more thing that you have to learn. One more thing that you have to learn is, see, I told that pollination will happen. Okay, I told that pollination will happen, but I did not. Yeah, I told you pollination will happen, but I did not tell you who are the agents of pollination. Now I know in smaller classes all of you would have studied agents of pollination, but still, okay. In exam they will ask you not agents of pollination, they will ask you different agents of pollination, what is the kind of plants or flowers that they are going to bear. Okay, so agents of pollination only 3 things you have to study. First, ನೀವು biotic, okay, and second thing is abiotic. Biotic and abiotic. Biotic and something that is living, right? So biotically you have to study about insects. Okay, because niಮಗೆ ಗೊತ್ತು insects are a major person who is going to do pollination. ಅದರಲ್ಲೂ ನಿಮಗೆ 2 categories ಇರುತ್ತೆ. One who comes for nectar, okay, and fragrance, like good fragrance. Okay, one more person will come for foul odor. Foul odor andre like something dead. Okay, dead kind of smell will come. For that bees will come. Okay, for that who will come? Bees will come. So nectar and fragrance, mainly your honeybees are going to be attracted. Okay, honeybees are going to attract. Foul odor, your beetles and flies are going to be attracted. Okay, flies are going to be attracted. So insects are there, students. We call this entomophily. Now abiotic andre the non-living things. So non-living alli nimage eradu ide. Okay, non-living you have 2 of them. What are they? Water, hydrophily, anemophily, wind. Okay, wind pollinated. So one is water pollinated, one is wind pollinated, but eitherly most common andre idu wind pollination. Okay, most common is wind pollination. Water pollination ಸ್ವಲ್ಪ rare ಆಗಿರುತ್ತೆ. Okay, because it's not easy to do water pollination, so it is going to be a little rare. Okay, little rare. Can you not understand my handwriting? Rare. Okay, this is going to be most common. Most common. Now, depending on the students, different types of flowers are going to be there. Different types, how they are going to bear. Like different types of flowers are going to be there. Clear? Yes. So, abiotic and biotic. Okay, biotic or abiotic. Now, insects is there, nectar and fragrance they come for. Foul odor is there, water and wind is there. Now, depending on yav tara agents. Okay, yav tara agents is there. Depending on that, pollination, like the flower modifications are going to be different. Now students, insect alli enagutte, normally neevu honeybee nodidre, this is your honeybee. Trust the process, this is going to look like a honeybee, okay? I know you people will be judging me now, don't judge, okay? Kind of. Okay, this is a honeybee. Now on the honeybee, what is going to happen? Let's say if there are pollen grains, the pollen grains are going to attach to the body, right? Like the fur, it is going to attach. Now this honeybee is going to go and sit on some other flower and it is going to dust this pollen grains there. Okay, so that is how this pollination is going to happen. Wind and water alli no difference illa. Water alli enagutte students, on the surface of water. Itara surface of water. Okay, itara surface of water alli pollen grains release agutte. Let's say this is the pollen grain. Itara pollen grains release adre, the pollen grains will be covered. Okay, one mucilaginous coat in the cover agirutte. Why? Because it should not sink in water and it should not get wet in water. Then the female flower, okay, the female flower, stigma iruttalla, female flower will be going near or taking it, something like that. Okay, windal normally what will happen? As the wind flows, the stigma and the pollen grains will go and it will go and fall on the stigma. Clear? Now, this is what is the different types of modifications that are going to be there. I hope this is clear to all of you. Okay, okay, very nice. Students, I'll get some water and come. Okay, I'll just get some water and come. I want all of you to wait. By then, you can copy down the notes. If you want, I'll keep the megasporogenesis slide because I know most of you. Students, notes, smarty students, it will help you a lot. KCT preparation time, NEET preparation time, these notes will help you. Okay, that's why I told you, come to my Telegram as well because all notes are available. So, I don't know which notes you will be wanting. This page irbodu. Okay, just make a note of this. By then, I'll just get some water. Done? Okay, continue madana. Starting with the first portion. Okay, pollination by wind is more common. It already ಹೇಳಿದ್ದೀನಿ ನಿಮಗೆ. More common. amongst the abiotic pollination. Wind pollinated also requires, ivaga nimige board exam ge CBSE students agirli, PUC students agirli, board exam they're going to ask you write any 5 features or write any 2 features that are shown by wind pollinated flowers, okay, or wind pollinated plants. So you have to write what are the adaptations that they have done so they can become wind pollinated is what you're supposed to write. Wind pollination also requires that the pollen grains are light, okay, pollen grains are light, non-sticky, so that they can be transported in wind current. So if the pollen grains are going to be heavy and sticky, ivaga wind bandre, can it fly? No. Now let's imagine on my hair I've put lot of oil. Now if the wind is flowing, do you think my hair is going to fly? My hair is not going to fly. Why? Because it is completely sticky, right? Oil is completely sticky agirutte. I think girls can relate more. Okay, girls can relate more how the hair gets very sticky. Okay, so that they can be transported in wind currents. They often possesses well-exposed stamens. As you can see, well-exposed stamens they are going to have and anthers they are going to have, so that the pollens are easily dispersed into the wind current. So this is your stamens. You can see they are well exposed. Why? So that as the wind flows, the pollen grains can be moved. Okay. Now, even the stigma is usually large, often feathery stigma, to easily trap the airborne pollens. Andre, in students, this pollen grains will be coming through the wind, no? Now imagine the stigma is this kuti. Now will the pollen grains land on the stigma? Ma'am, what are you saying, ma'am? It's not possible. Exactly. The pollen grains will not land on the stigma. So what do we need? We need proper feathery stigma which will catch hold of the pollen grains. Okay. Now wind pollinated flowers often have a single ovule in each ovary and numerous flowers. Okay. So what happened? They will have a single ovule most of the time. Okay. And they will have numerous flowers which are packed into an inflorescence. So students, it usually enik martarandre so that more pollen grains, okay, can be scattered at the same time. Because if the flowers are too tiny, it is too difficult for the wind to carry the pollen grains, right? It will be very difficult. So what they do, they come to a cluster of flowers. Okay. Now imagine madi on helicopter hogta ide. We want to convey some message to the helicopter. Naan ivaga obne itara hing madkondidre yaru nodalla nanna. Let's imagine I have called 10,000 people and all the 10,000 people are doing like this. Yes, definitely some speck you can see down from the helicopter, right? Example. Okay. Now during this flood in Kerala and all, during the flood enaito, people were putting lights and they were doing this, but when one person was doing it was not spotted. But when a group was doing, 5, 6 people, 7, 8 people was doing this, yes, they were able to spot those people above the terrace and they were able to save them. So aatarane illo chikku flower agidre, there's no way that the air will be able to take this pollen grain, so they will form a cluster. Okay. Now a familiar example is corn cob. The tassels you see, no, the ears. Okay, ivaga new NCRTL ears in the kottidare because it's not tassels. Okay, the ear you see are nothing but the stigma and style. Okay, so corn cob ear, not the tassel. The corn cob ear is going to be the stigma and style which wave in the wind and trap the pollen grains. Wind pollination is quite common in grasses. Important. Okay, wind pollination is quite common in grasses. Clear to all of you? Now, pollination by water. Okay, pollination by water. Now, pollination by water is quite rare in flowering plants and it is limited about to 30 genera. Okay, it is limited to how much students? 30 genera. Mostly monocotyledons. Mostly what? Monocotyledons. As against this, you would recall that water is the regular mode of transport. Okay, students, adike navu bryophytes na amphibians of the plant kingdom anta karitivi. Yake? Because ee bryophytes banbittu avrige neer illa andre on life cycle complete agalla. So amphibians aatarane land matte water eradu beku avrige complete life cycle complete madake. So that is why we call bryophytes amphibians of the plant kingdom. So ill nodi, you will recall that water is a regular mode of transport for male gametes among the lower plants. Groups such as algae, bryophytes and pteridophytes. Okay. It is believed particularly for some bryophytes and pteridophytes that their distribution is limited because of the need for water and for the transport. Andre il main problem enagutte students, ee water alli if they release the pollen grains, there are chances that the water cannons will carry them. So it is not 100% sure that they are going to meet the female flower and fertilization will happen. So kela flowers avaru ಏನು ಮಾಡ್ತಾರೆ, ಅವರು ಈ ತರ ವಾಟರ್ ಲೆವೆಲ್ಗೆ ರೈಸ್ ಆಗ್ತಾರೆ. ದಟ್ ಮೀನ್ಸ್ ದೇ ವಿಲ್ ಕಮ್ ಅಬೌವ್ ದ ವಾಟರ್ ಲೆವೆಲ್ and they will wait for wind or insect pollination to happen. This was a NEET 2023 question. So listen to me carefully. Examples of water pollinated flowers are Vallisneria and Hydrilla, okay, which grow in fresh water, and several marine seagrasses as Zostera. Not all aquatic plants use water for pollination. In majority of the aquatic plants such as water hyacinth, okay, and water lily, okay, the flowers emerge above the water and are pollinated by insects or wind. See what they do? In water lily and water hyacinth, the ಎರಡು species alli avaru water pollination ge they are not very comfortable, they do not believe in that. So what they do, they come above the water level. Okay, so what they do students, simple, il nodi, see this is the water, okay, these people with the help of long stigmas and stamens, they will come above the water level like this. Okay, they will come above the water level, and what will they expect? They will expect that wind or an insect comes to pollinate them. A wind or an insect comes to pollinate them. So, this question bandito, they had given in water hyacinth and water lily, what is the kind of pollination that they have. So, many students, what happened, because our water anta nodi confused nagi water pollination anta bardidare, but answer bandito insect and wind. As of the most of the land plants. In Vallisneria, the female flower reaches the surface of water by the long stalk. Okay, by the long stalk, and the male flowers or pollen grains are released onto the surface of water. They are carried passively by water currents. Passively by water currents, are they not like completely taking the pollen from here and putting it in America? Not like that. Passively they are moved by water currents, and I told you they will have a mucilaginous cover, so the wetting can be avoided. Okay, some of them eventually reach. See, some of them will eventually reach. Ella reach agala, but some of them will reach the female flowers and the stigma. Okay, in another group of water pollinated plants, the seagrass, what happens again? The female will come up. Now I told you there's a mucilaginous covering for these people. Okay, there's going to be a mucilaginous covering for these people. Next is going to be insect pollinated. Okay, here what is going to happen, students? Majority of the flowering plants use a range of animals as pollinating agents. Example, bees, butterflies, flies, beetle, wasps, ants, moths, birds, etc. And bats are the common. See, bats are the common pollinating agents. Okay, bats are the common pollinating agents. Among the animals, insects, in insects particularly, bees are the dominant one. Okay, bees are the dominant biotic pollinating agents. Even larger animals such as some primates like lemur, tree dwelling lizards, chameleons, idella ಸ್ವಲ್ಪ help ಮಾಡ್ತಾರೆ, because our body allu adu pollen grains attach agi they will move everywhere, so pollen grains transfer ಆಗುತ್ತೆ. Okay, lizards, garden lizards have also reported to be pollinators. Now look here, what is the speciality of insect pollinated flowers? Il nodi, first of all, insect na attract madbekandre aur large and colorful agirbeku. They should have nice fragrance, they should have honey, nectar should be there. If all these are not there, the honeybee is not going to come back, any of the insects are not going to get attracted. So majority of the flowers, they will have large, colorful, fragrant, rich in nectar. See whatever I told you. When the flowers are small, what happens? They form an inflorescence. Cluster form marbit, inflorescence ಮಾಡ್ತಾರೆ if the flower is small. Okay. Now, animals are attracted to flowers by color or by fragrance. The flowers pollinated by bees and beetles. ನಾನು ಹೇಳಿಲ್ವಾ, bees and beetles ಗೆ foul odor ಇರುತ್ತೆ. Okay. To sustain animal visit, ಅಂದ್ರೆ animals ವಾಪಸ್ ಬರ್ಬೇಕಂದ್ರೆ, that means they have to come back and visit. Flowers have to provide some rewards. Flowers have to provide rewards, just like how I tell you people that you have to like and comment, so I feel like coming back and taking your classes. If you don't show any interest, I'll be like, okay, leave anyway, students are not interested, why will I waste my time, right? So that's why I tell you, like, reward. So if you should come back to my classes every day, I should make sure that you understand everything. So when I am doing that, I expect that also from you, okay? So flowers have to provide rewards, okay? Rewards to the animals. The reward is going to be not fragrance and nectar, but the reward is going to be nectar and pollen grains, okay, are the usual floral rewards, For harvesting the rewards from the flower, the animal visitors come in contact with anthers and stigma. The body of the animals get coated with the pollen grains and they go and sit on another flower and the pollen grains are going to be distributed. Clear to all of you? Yes or no? Okay. Now students, illi on different type of example kodidare where you will be able to see something called as mutualism. Mutualism anta helbodu andre ನಾನು ನಿನಗೆ ಹೆಲ್ಪ್ ಮಾಡಿ ನೀನು ನನಗೂ ಹೆಲ್ಪ್ ಮಾಡು ಆ ಥರ ಒಂದು ರಿಲೇಷನ್ಶಿಪ್. ಓಕೆ, ಇಲ್ಲಿ ಎಕ್ಸಾಂಪಲ್ ಬಂದ್ಬಿಟ್ಟು ಒನ್ ಆಫ್ ದ ಟಾಲೆಸ್ಟ್ ಫ್ಲವರ್ ಆಫ್ ಅಮೋರ್ಫೋಫ್ಯಾಲಸ್. ಆ ಫ್ಲವರ್ ಹೆಸರೇನು? ಅಮೋರ್ಫೋಫ್ಯಾಲಸ್. ಈ ಫ್ಲವರ್ ಏನು ಮಾಡುತ್ತೆ ಅಂದರೆ ಪಾಲಿನೇಷನ್ ಇಸ್ ಎ ಡಿಫಿಕಲ್ಟ್ ಟಾಸ್ಕ್. ಓಕೆ, ಎವ್ರಿ ಫ್ಲವರ್ ಡಸ್ ನಾಟ್ ಗೆಟ್ ಪಾಲಿನೇಟೆಡ್. ಸೊ ಅಮೋರ್ಫೋಫ್ಯಾಲಸ್ ಫ್ಲವರ್ಗೆ there will be some insects, okay, some beetles, different types of beetles. They'll be like, I'll do one thing, I'll pollinate for you, but instead you have to take care of the eggs that I'm laying in your ovary or in the ovule. So ee tara one condition that you protect the eggs that I'm laying and I will do pollination for you. So ee tara one mutualism nadiyutte Amorphophallus. Second example bandu yucca plant. Yucca plant with Pronuba moth. Pronuba anta moth hesaru, yucca moth anta karitare. Adu same students, a moth bandu it lays the eggs in the ovules, okay, or in the ovary. So here you can see locules. Locules of the ovary, that means where the ovules are going to be present. Locules of the ovary. Now what is the plant's job? It has to take care of the eggs. And what is the moth's job? Moth has to make sure that pollination takes place. So this is like a mutualism. That is, I will help you, you help me, we both will be ever, forever happy. See, this is the image. This is the yucca moth. Okay, this is the yucca plant. And you will be able to see that these people are there. Here also you can see so many beetles around Amorphophallus flower. Okay, this is the actual image. Amorphophallus flower ಬಂದ್ಬಿಟ್ಟು ತುಂಬ ದೊಡ್ಡದು. Okay, almost 6 feet height. ಅದು ನೋಡಿ ಆ ಇನ್ಸೆಕ್ಟ್ ಬಂದ್ಬಿಟ್ಟು ಓವರಿಯಲ್ಲಿ it will lay the eggs. ಇಲ್ಲೂ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್ ಸೇಮ್ ಥಿಂಗ್. ಒಂದು ವೈಟ್ ಕಲರ್ ಯೂಕಾ ಮಾತ್. ಓಕೆ, ವೈಟಿಶ್ ಕಲರ್, ಫ್ಲವರು ವೈಟಿಶ್ ಕಲರ್ ಇರುತ್ತೆ. ಯೂಕಾ ಮಾತ್ ಏನು ಮಾಡುತ್ತೆ? It will go and lay its eggs in the ovule or in the ovary and it will tell the plant that please take care of it. I will make sure that pollination takes place. So this is the mutualism example. They might ask you what is mutualism and give me an example for mutualism. I hope this is clear. Okay, now we will do outbreeding devices. Okay, outbreeding devices andre nanu ivaga helidini students that if you are marrying inside the family, right, genetic problems continue ಆಗುತ್ತೆ ಅಲ್ವಾ. ಸೊ ಇಲ್ಲಿ ಪ್ಲಾಂಟ್ಸ್ಗೂ ಆ ಥರ ಒಂದು ಪ್ರಾಬ್ಲಮ್ ಇದೆ. ಕಂಟಿನ್ಯೂಸ್ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಮಾಡಿದ್ರೆ ಅವ್ರಿಗೆ ಡಿಪ್ರೆಷನ್ ಬರುತ್ತೆ. ನಮ್ಮ ಥರ ಡಿಪ್ರೆಷನ್ ಅಲ್ಲ, ಬೇರೆ ಥರ ಡಿಪ್ರೆಷನ್. ಓಕೆ, ಡೋಂಟ್ ವರಿ, ನನಗೆ ಥ್ರೋಟ್ ಇನ್ಫೆಕ್ಷನ್ ಆಗಿದೆ. ಸೊ ಅದಕ್ಕೆ ಸ್ಟ್ರೆಪ್ಸಿಲ್ಸ್ ಕಂಟಿನ್ಯೂಸ್ ಐ ಹ್ಯಾವ್ ಟು ಕೀಪ್. ಸೊ ಈ ಥರ ತುಂಬ ಪ್ರಾಬ್ಲಮ್ಸ್ ಇದೆ ಸ್ಟೂಡೆಂಟ್ಸ್. ಟೀಚರ್ಸ್ಗೆ ಮೇನ್ ಪ್ರಾಬ್ಲಮ್ ಥ್ರೋಟ್ ಟೈಪ್ ಪ್ರಾಬ್ಲಮ್. ಓಕೆ, ಸೊ ಔಟ್ಬ್ರೀಡಿಂಗ್ ಡಿವೈಸಸ್ ಅಂದರೆ ಸ್ಟೂಡೆಂಟ್ಸ್ ಬಿಕಾಸ್ ಆಫ್ ಕಂಟಿನ್ಯೂಡ್ self-pollination. Continued self-pollination leads to something called as inbreeding depression. Inbreeding depression. Okay, ಈ inbreeding depression avoid ಮಾಡೋದಕ್ಕೆ plants have some techniques. First of all, self-pollination ಹೆಂಗಾಗುತ್ತೆ? ಇವಾಗ ಆಂಥರ್ ಇದೆ, okay, and stigma is there. Pollens will fall like this. So same plant, continuous self-pollination will happen. So to avoid to avoid inbreeding depression, plants have made some outbreeding devices. ನಿಮ್ಮ NCERT ಅಲ್ಲಿ 4 devices ಕೊಟ್ಟಿದ್ದಾರೆ. So we will also study the 4 of them. First one ಬಂದ್ಬಿಟ್ಟು students, formation of unisexual flowers. Unisexual flowers andre eno, in one plant, okay, in one male will be there, in another female will be there. Okay, unisexual flowers. So we can say staminate flower and pistillate flowers. Just like papaya. Okay, that means in one plant you will see only the male part, in other plant you will only see the female part. Okay, formation of unisexual flowers. ಈ ಟೆಕ್ನಿಕ್ ಇಂದ ಏನಾಗುತ್ತೆ? ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ಗೆ ಚಾನ್ಸೇ ಇರಲ್ಲ, ಬಿಕಾಸ್ ಒಂದು ಪ್ಲಾಂಟಲ್ಲಿ ಮೇಲ್ ಇರುತ್ತೆ, ಇನ್ನೊಂದು ಪ್ಲಾಂಟಲ್ಲಿ ಫೀಮೇಲ್ ಇರುತ್ತೆ. ಇಲ್ಲಾಂದರೆ ದೇ ಜಸ್ಟ್ ಫಾರ್ಮ್ ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್. One of the parts only will be there, neither the male or neither the androecium or the gynoecium. Now there is no way that it will disturb or it will form self pollination or do self pollination. So aa tara on hopefully they will form unisexual flowers. Okay, second one enandre students, second one is changing the receptivity or we can even say maturity, asynchronized receptivity or we can say maturity. ಇದೆಲ್ಲ ಏನಾಗುತ್ತೆ ಸಿಂಪಲ್, ಇಲ್ಲಿ ನೋಡಿ. ಸೊ ಆನ್ಸರ್ ಇಸ್ there. Okay, stigma is there ಅಲ್ವಾ. ಸೊ ಇನ್ ಸಿನಾರಿಯೋಸ್, ಓಕೆ ಒಂದು ನಿಮಿಷ, okay, look here, what is going to happen? Normal scenario students, normal scenario ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಆಂಥರ್ ಆ್ಯಂಡ್ ಸ್ಟಿಗ್ಮಾ ಒಂದೇ ಟೈಮಲ್ಲಿ ರಿಸೆಪ್ಟಿವ್ ಆಗುತ್ತೆ. That means anther also will mature at the same time, stigma also will mature at same time. Okay, so the pollens from the anther will land on the stigma and they will continue fertilization. But ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, let's imagine anther ಇವಾಗ receptive ಆದರೆ stigma will not become receptive. So even if the pollen grains are released, okay, it is of no use. It is of no use. Clear to all of you? So first scenario dalli anther and stigma ಒಂದೇ ಟೈಮಲ್ಲಿ ಮೆಚೂರ್ ಆಗ್ತಾ ಇತ್ತು. ಸೊ ದ ಕಂಟಿನ್ಯೂಸ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಯೂಸ್ ಟು ಹ್ಯಾಪನ್. ಇಲ್ಲೇ ಏನಾಗ್ತಾ ಇದೆ, ಆಂಥರ್ ಇಂದ ಸ್ಟಿಗ್ಮಾ, ಆಂಥರ್ ಮೆಚೂರ್ ಆಯಿತು ಬಟ್ ಸ್ಟಿಗ್ಮಾ ಮೆಚೂರ್ ಆಗಿಲ್ಲ. ಸೊ ಅದಕ್ಕೆ ಏನಾಗುತ್ತೆ, ಆಂಥರ್ ಇಂದ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ರಿಲೀಸ್ ಆದ್ರೂ ಸ್ಟಿಗ್ಮಾದಲ್ಲಿ ಫರ್ಟಿಲೈಸೇಷನ್ ಕಂಟಿನ್ಯೂ ಆಗಲ್ಲ. ಸೊ asynchronized receptivity, that means both will mature but at different timings. Okay, next one is going to be, ಇಲ್ಲಿ ಡ್ರಾ ಮಾಡ್ಬೋದಾ, ಚೇಂಜಿಂಗ್ ದ ಲೆಂತ್. ಓಕೆ, ಲೆಂತ್ ಆಫ್ ಆಂಥರ್ ಆ್ಯಂಡ್ ಸ್ಟಿಗ್ಮಾ. ಫ್ರೆಂಡ್ಸ್ ನಾರ್ಮಲಿ ಇಲ್ಲಿ ಡ್ರಾ ಮಾಡ್ತೀನಿ. ಓಕೆ, ನಾರ್ಮಲಿ ಏನಾಗುತ್ತೆ, ಇದು ಸ್ಟಿಗ್ಮಾ, ನಾರ್ಮಲಿ ಈ ಥರ ಇರುತ್ತೆ. Normal situation, so the pollen grains can fall on the stigma. Correct. Now what they will do, okay, this is the scenario, normal scenario. Okay, now what the plant will do, plant will change the length of one of them. Neither it will make the stigma longer, there is the style longer, and it will make the anther smaller. So they do something like this. Ivaga, is it possible? Ivaga, it's not possible. Like now it's not possible. Why? Because the height is different. Okay, heterostyle height is completely different. Clear to all of you? Yes or no? Done? Illi barithini ಒಂದು ನಿಮಿಷ. Outbreeding devices, they will ask you to write. Okay, outbreeding, yeah, outbreeding devices. Clear? Now let's just select this. Okay, so outbreeding devices. Okay, it's very important for you to remember. Now last one, what they will do students, they will do something called as incompatibility. Okay, what is that called? Incompatibility. Incompatibility andre stigma will reject the self pollens, that means the pollens that has fallen on them. Okay, so it will self incompatibility. That means you are going to reject all the proposals that is coming from inside your family. You are going to reject all the proposals that is coming from inside your family. Clear? Whoever comes like inside your family, you are going to reject them. That means what is going to happen? Illnodi stigma. Okay. Now pollen grains are fallen. Okay, blue color pollens ella bere kade pollens. Okay, pink ella self pollens. Okay, same plant inda bandirodu. Ivaga this instruction is given to the stigma telling that you do not accept the self pollen, you only accept other pollens. Okay, so what is the stigma going to do? It is going to completely reject the self pollen, and it is going to allow the other pollens to germinate. Clear to all of you? So 4 steps they are doing is unisexual flowers, asynchronized receptivity, length of the anther and stigma will be changed, and self incompatibility. Okay, self incompatibility. Clear? Okay. So that is your outbreeding devices. You'll notice NCRT. This is a genetic mechanism and prevents self-pollen, okay, from the same flowers or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or the pollen tube growth in the pistil. Another device to prevent self-pollination is production of unisexual flowers. Unisexual flowers prevents self-pollen, okay, self-incompatibility. If both male and female flowers are present on the same plant such as castor and maize, monoecious, it prevents autogamy but not geitonogamy. In several species such as papaya, male and female flowers are present on a different plant, that is each plant is neither male or female. So, dioecious, okay. Dioecious, this condition prevents both autogamy and geitonogamy. Delete martini. Next is pollen pistil interaction. Do you all want a break? Do you all want a break? If you want a break, just let me know. Want a break? Chalo, take a break.
their, war practices or, the pre-battle training. Will that be okay? No, ma'am, it won't be okay. But why are you talking about that question? Very simple, students. Colon system. Any colon from anywhere can come to follow the sigma. ಇವಾಗ ಸಿಗ್ಮಾ ಹೆಂಗ್ ಗೊತ್ತು that this is the correct colon ಅಂತ ಒಂದು ಟೆಕ್ನಾಲಜಿ ಇರ್ಬೇಕಲ್ವಾ, ಒಂಥರ ಮೆಕ್ಯಾನಿಸಮ್. ಸೊ ಅದಕ್ಕೆ ನಾವು ಕೋಲನ್ ಟೆಸ್ಟೆಡ್ ಇಂಟ್ರಾಕ್ಷನ್ ಅಂತ ಕರಿತೀವಿ. ನಾವು ಈ ಕೋಲನ್ ಟೆಸ್ಟೆಡ್ ಇಂಟ್ರಾಕ್ಷನ್ ಏನಾಗ್ತದೆ ನೋಡಿದ್ವಿ. ಸೊ ದಿಸ್ ಇಸ್ ಯೋರ್ ಸಿಗ್ಮಾ. ನಮಗೆಲ್ಲ ಗೊತ್ತು ಸಿಗ್ಮಾ ಇಸ್ ದ ಲ್ಯಾಂಡಿಂಗ್ ಪೋರ್ಷನ್ ಫಾರ್ ದ ಕೋಲನ್ ಅಲ್ವಾ. ಸೊ ದಿಸ್ ಇಸ್ ಯೋರ್ ಲ್ಯಾಂಡಿಂಗ್ ಪೋರ್ಷನ್. Okay, this is your landing page. Now just imagine that one colon has formed. Okay, one colon has formed. This is one colon. Okay, this is another. Now I'm going to do marriage proposal there. Now if a boy and girl meet, the boy will tell, I will take you for Europe trip for honeymoon. I will make sure that, I will let you discuss them like this. He will tell that I have girls, I have, I have there's a laparoscopy, I have all the dents, everything to them. Let's say another person, doctor, is coming. He'll be like, don't worry, I'll take care of the parents, I'll take care of yourself, I'll make sure that every checkup is done properly, I will make sure that the health is proper. Let's say an engineer comes and he'll be like, don't worry, I will take care because I'm very financially well to do, because I have a lot of clients and everything. So I'm well to do and I'm working in a big industry or whatever. Okay. Now, whom will the girl select? Completely depends on the girl. Alva. If the girl wants a person who is, having all the expensive cars, I'm not saying doctors in Lanka and engineers in Lanka, let's imagine, let's just people business people will have. Okay. So, now the girl will decide. Alva. So, after in this golden cases, ಮತ್ತೆ sigma, among this, sigma will decide who is compatible candidate. This word we use even in relationships. You are not compatible candidate. That means what? That means you are not matching my type. Correct? So here, same thing. Compatible. Colon system interaction talks about if colon and sigma is compatible to each other. So compatibility means, listen up. First, they are going to have something called as chemical dialogue. Chemical dialogue. So says once the colon range lands on the sigma will release a chemical which allows the colon range to continue the colon tube germination. Okay, so our chemical dialogue, on the marriage proposal, first I will matlada that I like this, this is my passion, this is my hobby, blah blah blah. So after that only the communication of the marriage proposal will come. So after that only first some chemical dialogue exchange after that. Then they will decide that okay, we have to go further with it. So after the chemical dialogue, the second step that the sigma is going to do, let's take example, sigma rejected this fellow and rejected this fellow. It has accepted the doctor. Okay, she accepted the doctor because she is also a doctor. So here what is going to happen? I'll show you. So here slowly you will start seeing colon tube growth. So the second step that is going to happen here is going to be colon tube germination. What is going to happen? Colon tube germination is going to happen. So this is called as colon picture interaction. Now I'll show you an image how it looks like. So students ನೋಡಿಲ್ಲಿ colon ಗೆ from stigma ಮೇಲೆ settle ಆಗಿದೆ. Okay. ಅಲ್ಲಿಂದ ಏನಾಗ್ತಾ ಇದೆ growth ಆಗ್ತಾ ಇದೆ. Okay, small ಈ ತರ growth ಕಾಣಿಸ್ತಾ ಇಲ್ಲ growth ಆಗ್ತಾ ಇದೆ. So you can see how the stigmatic process. So stigma and colon veins are exchanged the chemical dialogue. After that stigma has decided, okay, this colon vein is fine, we will make this growth. So after that, can you see how the colon tube growth is happening? Okay, so from here the colon vein is fallen, from here colon tube is growing and it is flying to see. It is entering into the ovules and where it is going into the. So this is how in this process. Okay, so NCRT diagram ನೋಡಿ. What is happening? Plasma membrane is synergic. What is synergic? One main gamete and vegetative nucleus to become attachment is going to guide them to enter into the reach the egg. Okay. Now here something different is going to happen. So what will happen? Let's take example. So ಈ ಸಿನಾರಿಯೋದಲ್ಲಿ ನಮಗೊಂದು ಡಾಕ್ಟರ್ ಇತ್ತು, ಇಂಜಿನಿಯರ್ ಇತ್ತು ಅಲ್ವಾ? Let's take this scenario where the doctor was there. Okay, this is the businessman. Okay, this is the doctor who grew the colon tube. Okay, then we have the engineer. Now engineer is like, no, I came for this marriage, even I want to be a part of it. Same problem, right? So since each of the situations are there, when I taught you in the starting of the chapter that there's something called as pre-cell shape. Right, pre-cell shape. Normally students, in angiosperm, there's something called as two-cell shape. Two-cell stages. Two-cell stages, very simple, okay? One vegetative nucleus is there, okay, vegetative cell. ಮತ್ತೆ ಮೇಲ್ ಗೆಮಿಟ್ ಒಂದೇ ಒಂದು ಮೇಲ್ ಗೆಮಿಟ್. Okay, but in some situations, okay, this happens in 60% of the angles. Okay, 60% of angles. Second scenario where again this one. Second scenario what happens in 40%. In the rest of the 40% or let's say of the world, ಇಲ್ಲೇನಾಗುತ್ತೆ there's something called as three-cell shape. So halfway coming to it, generative cell will be speaking. So generative cell will be like, you know what, I will undergo one more investigation and I'll come to know it. In case that happens, let's imagine engineer and doctor wants to be part of this marriage and we have only done that right. So in this problem, two grooms and one bride in this problem. So we have to arrange one more bride. I'll tell you who's going to be the bride. So in angiosperm 40% moves three-cell stage. Three-cell stage means in one vegetative cell is there because vegetative cells divide in this. ಇಲ್ಲೇನಾಗುತ್ತೆ two male gametes. Now since the two male gametes ನಾವು ಹೆಂಗ್ accommodate ಮಾಡೋದು, ಹೆಂಗ್ ನಾವು fertilization ಮಾಡೋದು ಅಂತ ಒಂದು ದೊಡ್ಡ ಕನ್ಫ್ಯೂಷನ್ ಇದೆ ಅಲ್ವಾ? So ಅದಕ್ಕೆ all of you listen to me carefully. ಏನಾಗುತ್ತೆ, see what is going to happen. I will draw it in circles. Okay, this is one male gamete. Okay, this is the second male gamete. Okay, different color I'm not drawing. Male gametes are already same. Okay, this is the egg. Okay, and now we have to find a second bride, right? One bride we have. Okay, one bride we have. We don't know where the second bride. So second bride is going to be the polar. What is it going to be? The polar nuclei. So one of the male gametes will fuse with here and one of the male gametes will fuse with the polar. Okay, draw it in circle for it. It will fuse with the polar. Okay, so it's here these two plus and these two plus. That means here male gamete. Okay, here also male gamete. Here the male gamete is going to fuse with the egg. Okay, process we call as triple fusion. We call it what? Triple fusion. Triple fusion. So, one situation where we got male gametes plus eggs, we got a cycle. That process is called twin gap. Male gamete, second male gamete, the engineer fellow who made a problem. First of all, why did engineer come? Because the generator cell decided to do mitosis. Generator cell was like, wait, I want to do mitosis. So, friends, ಇಲ್ಲಿ ಈ process ನಡೀತಾ ಇದೆಯಲ್ಲ ಇಲ್ಲದು ಇದು. So, in between here, remember one tube nucleus into there, which is going to lead the path, and with that, the one male gamete is going to be. But generator cell decided that it will do mitosis, so it will also share and male gamete committed. One male gamete will go and fuse to the egg, one male gamete will go and fuse to the polar. Okay? So, ಇಲ್ಲಿ ಈ process ಅಲ್ಲಿ ಅದಕ್ಕೆ ಏನು ಫಾರ್ಮ್ ಆಗುತ್ತೆ? We will get one male gamete plus polar nuclei is called as primary endosperm cell and triple fusion. Now listen here, in your NCERT you don't get confused. I'll tell you exactly what this primary endosperm cell is. So this is the primary endosperm cell. Okay. This is the primary endosperm cell. Okay. In the cell field, there is going to be primary endosperm nucleus. Okay, can you see the box? Yes. So, remember the labeling. This is going to be PET. Okay, and this is going to be PEN. PEN is primary endosperm nucleus. Okay, nucleus. Clear to all of you? So, this is what a primary endosperm cell. So, don't get confused with PET and PEN. Don't get confused with PET and PEN. Here, now let's show all of you. Double fertilization difference. The ultimate, what is going to happen? Double fertilization. What two steps are happening? Double fertilization means syngamy plus triple fusion. Syngamy plus triple fusion is called as what? Double fertilization. It's called as what? Double fertilization. Double fertilization. This is what happens. Okay? So that is why we call it double fertilization. This only happens in what stage? This only happens in a 3-cell ancestor. Remember that. This double fertilization happens with a 3-cell ancestor. That is in 40% of them. Okay, in 40% of them. Now, remember triple fusion again, the PEC form of it. This PEC is going to ultimately become what? This PEC is going to ultimately form the endosperm. PEC is going to ultimately form endosperm. Okay? And the zygote that we got from here. Zygote that we got from here is going to ultimately form embryo. Now this endosperm, students, is going to provide nourishment to growing embryo. So you tell me in the chat, sex form agega first in the zygote form agega first. Yeah, first form agega. Ma'am, common sense. Obviously, endosperm has to form first because that is the fellow who is providing food for the growing embryo. So obviously he should be formed first, this fellow should be formed later. Okay? So question with the example. So friends, let's tell the thought. This is because ನಮ್ಮ polar nucleus one cell but ಆದರೆ ಎರಡು nucleus ಅಲ್ವಾ. So male gamete N, okay? ಇಲ್ಲಿ ನೋಡಿ N. In egg N ಆಗಿದೆ. Yeah, half by 1. But polar nucleus ಅಂದರೆ 3N. So that means the effect for the endosperm is going to be 3N. Triplet. That is why we say what triple fusion. Okay, triplet this is going to be. So here also your endosperm is going to be triplet. Okay, your embryo is going to be triplet. Clear to all of you? So this fellow is going to provide nourishment to this fellow. So So it is understood that this endosperm will have more signature, yes, then cytoplasm will be there just like how we studied in generative cells, correct? So this is what is there. Okay, yes, clear to all of you? 1 second. ನಾನು ನಿಮಗೆ ಕೋಲಿನ್ ಗ್ರೀನ್ ಡಯಾಗ್ರಾಮ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದೀಯಾ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. One second. Okay, I have to teach you actually one more concept, viability concept. Okay. Sorry, second. Okay, done. So friends, ನಿಮಗೆ ಒಂದು topic had to teach which I left in last class. Okay, I thought I taught the people that, so I'll teach you that after. Okay, so friends, this will have both signature, then cytoplasm, right? This will have to have the food to give the growing embryo. Okay, all the food that you have to give to the growing embryo will be with them. So they have to provide the nourishment for the growing embryo. So this is why we call it triple C. Okay, one more topic later, I'll teach you the colon grain part. I think this we have completed, double fertilization, differentiation, 2 years. Okay, done. Now one more condition is one more, one more thing that you have to study is artificial hypertrophy. Now let's say I'm a farmer, okay, and I got to know that someone is very nice, a very rich person is very nice, and they need a lot of flowers. So ನಾನು specific ಆಗಿ hybrid flowers ಇಲ್ಲಾಂದ್ರೆ special type of flowers ನಾನು I try to follow, ಅಲ್ವಾ? So now for that to happen, let the nature decide which are the pollen grains that are going to fall in the flower. Now in such situation, it becomes very difficult for me, it becomes problematic for me.